So this was kind of the eye opener. This is the field where we put our first tile line in, and where that query B is, that's where the tile line follows, and then kind of out of that lateral draws query A. So query A 254, query B 287, but there was a 70 bushel difference on just going from Corteva to a Bayer number there. So trying to figure out where that came from, because I can't find the file for it, but the fertility, you would swear, is backwards. There's more fertility in query A than there is query B. There's more nitrogen, more P, more K than query B. So the yield doesn't follow the fertility. What it does follow is the biofertility. So this is the microbiometer. So this would be further away from the tile line. There's our 65 colony forming units. Fungal bacteria ratio of 0.1 to 1. We're on top of that tile line, 34 to 66, 0.5. What we're shooting for is a 1-to-1 fungal to bacteria ratio, but that's where, I understand tiling's expensive, and like in our hills, I'd like to try it once, but there's a reason why these guys are tiling every acre in Iowa, and that trend's moving. It seems like every year. I think 5 or 6 years ago, they were all west of Fort Dodge, and now they're 20, 30 miles or east of Fort Dodge. And this year, I had went out down Highway 20 for something, and yeah, there's tile plows going up and down these hills, 20, 30 miles west of Fort Dodge. And I talked to a guy picking up, we had to get a bin unload conveyor out in I can't remember. It's east of Webster City, and I was seeing all these tile plows. It was like 5 miles, and there's a crew here and crew there and crew there, and they're just going up and down the hills. And I asked the guy, I said, "Is it really worth it tiling?" I said, in my area, that's considered crazy. And he goes, "Well, it's been proven enough that it's you're crazy if you don't do it." I was like, "Well, what's it cost? This guy owns some ground. What's it cost you per acre?" And he's like, "Oh, it's costing around $1,300 an acre, on 60-foot centers." I was like, "Oh, well, that's less than what we charge." But why do you do it? Well, it pays itself off in 5 years. Within 5 years, and he goes, our APHs go up, and that's why they're starting to grow a lot more corn. And it's like, there's no need for drainage. So that's where this is kind of starting to support oxygen in the soil. We're able to hold on to the nutrients. If we're able to build the soil structure with the fungal, it makes the ground more permeable, more oxygen gets into the soil, and it actually holds on to more water. So it's able to soak in fast enough that it doesn't saturate and then wash all your nutrients off. This is what a root system can do with a corn plant. Bex is doing a lot of research on different root structures, but when we're digging up our roots, we're only seeing to the top bar, when we're taking a spade. But last year, our moisture probes, the deepest the corn roots got was 18 inches, and then we actually irrigated just shy of what we did in 23, trying to get that grain fill. And I'm kind of wondering, if you chop off, the corn roots, 18 inches, this is a 4-foot tall box. What are we leaving on the table for nutrients? If we have saturation, the corn didn't have a need to root down that deep. So getting air in the soil, saturation, and having that biology work for you, I think that's kind of where it fell short last year. Another thing that I'm finding with the biology, this is an Earth Optics result on a field that Dad, you haven't really put anything into this field for 15 years. No lime. The pH was way off. What we did in this little 4-acre patch. The field that we were in was right here. And this little patch, you know, trying to talk him into, we need to pattern tile some of these bottom ground. I think it's going to work. And so 5-acre patch, we decided we're going to pattern tile that and see what happens. So that has been, with this soil sample, it's been in for 3 years, but both sides of the dredge there, both fields have been managed the same. So seeing these differences, you know, we've got plenty of phosphorus, you know, 190 some parts per million. On the other side, still plenty high, but drops off, you know, 58, 35, you know, potassium, you know, everything on top of those tile lines, you know, both fields have been managed the same, other than there just being plastic pipe put underneath. I've never, you know, we haven't even seen water run out of that outlet. And you know, it was a field that never needed to be drained. There's a low spot, you know, in the driveway where there's compaction, but just seeing 3 years of that much of a difference between both sides of the dredge. It's same dirt, and the only difference is there's pipe underneath that 5 acres, and nothing's been done to the other side.
So all you think it's doing is oxygen coming through the pipe?
I think it's just oxygen, and it could be that the water's making it to the pipes, but it's, you know, leaching out of the tile line before, you know, because it's right next to the dredge too. It's just we have all that access, and the way I'm picturing it is water goes through that, when water goes through that soil profile with rain, there's got to be oxygen attached to that. You know, there's you know, rain's aerated too. So if we're able to let that water go down instead of pond and then just slowly leach, if we can give that an avenue to go. And
we put up a solar panel and like a little 3 horsepower pump to pump water out of the dredge into the tile outlet. We've got a structure there that we can pull a plug, but once it gets so high, it'll run over and go out the outlet. We've never been able to fill that structure with all the pipe on just 5 acres, and I think when the pump is running, it's pumping approximately 60 gallons a minute. But we've never been able to get the water level inside that structure more than a foot off the bottom, which means it would be about 3 feet below the surface. So, don't know if that has anything to do with it either, but
yeah, well, and 22 was a super dry year, and that was that the second year that was in, or was that the first year? Anyway, it was really interesting to see, you know, where we tiled it, there was 75 bushel corn in between the tile lines, but the 3 rows that the tile lines were right next to, every plant had 2 ears on it, and, you know, just counting rows around and stuff, there was 400 bushel corn on top of the tile line, 70 bushel corn in between, and it was dry. Well, we were pumping water back in, but that just keeps where I keep diving down the rabbit hole of what if we can replace these pivots. When we were putting it in, you know, we want to have a restrictive layer. One thing we learned was we put the tile lines too deep. We ran 3 foot, I think. We want to be closer to 2 feet deep, closer together, more pipe, to have more even distribution. But yeah, just seeing the, you know, the ground change, you know, that stark contrast.
So how close do you need to have pipes?
It needs to be half the distance of what it requires to drain. So in that Zook soil, in Zook we want to have our pipe 30 feet. We want 30-foot spacing. If we're going to sub-irrigate, they got to be every 15 feet. But where, and that, you know, that gets pretty spendy, but so does a pivot and the maintenance on that. So when we were digging the holes to connect the pipes, we have about it was right about that 3 foot mark. We've got 3 foot of black Zook and then it was like hitting concrete because it was dry when we put it in, and there was clay. We're still behind.
Zoo.
Are they gonna bring it down? Okay. Free cookies.
Free cookies or not?
So if you burn the 15 feet, you use smaller, right?
Yeah, we do. We'd use 3-inch.
But that was kind of the biology thing. Doesn't need tile, doesn't need drainage, but for some reason we're seeing the differences there. Last year, I'm doing the same thing this year. I haven't been as on top of it as I was last year, but with that Chrysolabs probe, I was mapping, trying to map the nutrient uptake through the soil. And so planted April something. Yeah, it's still fuzzy on that screen, but kind of showing the growth, through with the dates. So we're June, as soon as we kind of hit rapid growth V6, June 5th, we start seeing our nitrate levels plummet as that plant's trying to stretch out. The blue line, I had a number in my plot that did not have any seed treatment on it, did not have we didn't run any, we were running hopper throttle on the seed, and then in the field, we had it all treated, Invita on the seed, hopper throttle, talc. And so I was just curious to see, can we actually map and see these biologicals actually work? And so I find it interesting, the untreated nitrate, and you can see when I ran the pivot and added more nitrogen to it, 180 pounds total on it. But during that grain fill period, the untreated plummeted and the treated maintained and actually increased by the end of the year. So to me, that was enough. Okay, these biologicals actually do something. What blew me away, and where I'm really cautious about, like the phosphorus, and I'm going to get a soil map or soil test done with Earth Optics. I'm going to find out I just depleted all my phosphorus.
Joe, would you say it's more of a regulator because enzymes bug what nutrient they need?
Yep. So I think with the with the nitrogen, I think the plant was maybe there was more mineralization where the plants in the untreated were demanding that for the grain fill, and because this is just nitrate, so I don't know what that's all that probe measures in. So this is plant available. There was more plant available nitrogen at the end of the season with the treated than the non-treated. So now there's more nitrate out there. So I'm curious to see how the beans, where these samples were taken, perform versus where I put 120 pounds of nitrogen, and hopefully was able to make the most out of the biological and save the most cost on the nitrogen if that comes across on the soybeans. Because my big thing with soybeans is there should be money made in it when we know that when Randy Dowdy proves that we can grow 200 bushel soybeans, same thing with David Hula, these guys are putting, $2,000 an acre, to push the yield on this, but it's like, how hard do we have to push it, or do we even have to push it at all to get half of that? Can we save money, get half of what these record guys are doing, and be more profitable? So like with the soybeans, I've always been disappointed with soybeans in that field. White mold's been a struggle, but even before white mold, we'll have chest-high soybeans loaded with pods, count 200 plus pods on the plant, go through with the combine, we're combining 55 bushel soybeans, looking over the combine for where the hole's at, and finding that more than half the pods were aborted. Because like looking out there today, so it's like I don't have my hopes up. I'm excited for that soybean field, seeing that some of these treatments are working, and especially with the nitrogen. A guy told me when I told him that story, he goes, "How much nitrogen do you put on your corn?" Well, 200 pounds. He goes, "Do you need 200 pounds?" I don't know. Well, how much nitrogen is left over? Are your beans nodulating? Yeah, there's some nodulating, and we're looking at him. He's like, well, he goes, that's not nodulation. He goes, he said it's either sulfur or nitrogen that the beans are running short on. And he said that he would bet quite a bit of money that the beans are falling short of nitrogen at the end of the year because they've got plenty of nitrogen up front. It doesn't send the signal to the biology to start fixing N. It doesn't have nitrogen to finish out the end of the year. And then with that falling short, it doesn't have enough to fill the grain. And then tall beans, lots of fuel burned, and nothing to show for it. So last year, this is the P1 and P2 phosphorus. Blue and orange is untreated P1 and P2. Treated P1, P2 is gray and yellow. Started with 81 parts per million. This would be last year, so second year of not putting any fertilizer on. I think the first year started with 40 parts per million. So second year, no MAP, started with 81 parts per million on the untreated P2, ended up with 68. So there was some removal. The treated P2 started with 77 parts per million, ended with 90 after the crop was taken off. And the corn was yielding 290 to 311 where I took that corn off. So that's something that doesn't make sense. Crop removal. How many pounds of phosphorus does it take, to grow 300 bushel corn? How did I end up with more? It's working. I'm going to keep going. So in that soybean field, the plot is in this corner. This is the yield map from last year's corn. The plus sign, I got 180 pounds of total nitrogen, 140, 160, 140, and 120. So a couple things that stood out. 140 won it, but there's a good green strip where I had 180. And this is this was the fungicide trial last year. Biomenum, fungicide enhanced, regular fungicide, and then I threw all of it together. So when I saw this map, I was really discouraged and thought, oh, well, I was really hoping Biomentum would work, but it didn't. And then going through that same area with the combine, why do we see 300 bushel corn? Doesn't make sense. And then this year's soybeans, I didn't get a chance to get the map, but this year's soybeans, I was able to see this whole line this year. So that's where being patient with these biologicals, a lot of times, we're so used to everything being so instant. If you're deficient on nitrogen, you throw nitrogen out there, you expect it to work right away. You don't expect to throw it out there and then see the benefit from it next year. But on this side of the field, I think also a contributor is this is where I did my trial with Biomentum.
Jake, I had a question. What's the cost of Maris Neo versus Biomentum?
Last year it was $18.50 for Miravis Neo plus the $12.50 drone, so $30.31. And I sprayed 16 ounces of Biomentum, and that was $3 plus the drone.
And secondly, why is the all less yield than just a Biomentum?
Different hybrid. Yeah, there was a hybrid change there.
With the biological, it takes years to screw it up. You can't fix it in one.
Yeah.
So that's no different than no-till. You don't expect no-till to benefit the first year.
No. And I'm kind of thinking, I think it's I'm trying to collect all sorts of data from all different sources. Let's see if I can pull this out without breaking anything. So I like hearing what Verlyn has to say. I like hearing what the universities like to say. I like hearing Glenn Ravenberg and Tom Dykstra. Not everybody's got the perfect answer, but there's a lot of guys in different circles. So I think with where our ground is at, the way I understand it, this is peak peak efficiency of what we use for nutrients, and where I think our ground is at, especially seeing, after 2 years no MAP and seeing 90 parts per million of phosphorus. I think our fertility is over the curve, and it's going to be it's just going to take time to get back to that peak efficiency. If you're a guy that if we do the Earth Optics test and you've got good biology, good fungal to bacteria ratio, and it's a lot easier to go this way. It costs dollars, but you're going to see more of an instantaneous return on it. Is that the subway? Yeah. Thank you. Where, we're seeing nutrient tie-ups, we're seeing imbalances. This just takes time to go backwards. It's a lot easier to build forwards than it is to go backwards, and so that's what I'm excited about, and hoping that we see the benefits come back on. So I've been walking through the field looking at it like a hawk, even though the Earth Optics test said not detected. I planted that soybean plot over the worst acres for white mold, and there are 3 numbers in that plot that says do not plant on white mold acres. So went down every single one of those numbers, thinking if it's here, it'll show up in these numbers. Walked right down to the heart of the worst spot in the field, peeled the beans back, saw white mushrooms, started freaking out. But one thing about white mold is soybean is the only host for white mold. Dead soybean residue. It can only, it infects through the dead flowers. And then that's how it infects.
Ain't the mushrooms different for white mold than just how's up?
Yeah, they're kind of similar. I mean, they're not like fuzzy looking like that.
Well, upside down in the row, kind of.
Yeah.
Yep.
And they got like a dark, pin now. But just looking it up and seeing white mushroom. So pulling it up, and I'm finding that this is growing in the corn residue. I found a corn cob, and I wish I took a picture of it. But it was a corn cob, whole corn cob that came out of the combine, and the entire cob is just covered in those mushrooms. And I've never seen anything like that, and I sent the picture to Verlyn saying, is this something from Biomentum? Because either I haven't been that observant on just natural fungi and bacteria breaking down corn residue. And he said, yeah, that's most likely one of the fungal strains in Biomentum.
Never crop.
Yeah.
Harvested.
Yeah. I don't know if they're edible. They should be. But in that picture right there, this was on top of the ground, in the canopy, and there's an earthworm, living in a corn stalk. So that's where I'm really excited to see, the continual use, because these acres here saw a quart last year and saw just shy of 3 quarts this year. And the benefit with Biomentum, unlike Envita, you use it. It's not a native species. It grows with you during the growing season. It freezes, and Vita's dead. Pivot Bio's dead. A lot of these other residue digesters, looking them up, they're not native to our area. They can't overwinter. They can't survive freezing temperatures. Verlyn's stuff is accumulative. So it's like you add this, the only way you're going to lose it is if you dump fungicide on it and hurt it chemically. So it's neat seeing new life. In the cornfield, this is the soil samples. Actually, I'm gonna break. Let's grab some lunch, because I know you guys are hungry. So I just have a few more slides and kind of my theories of what's going on. So this is a soil test taken July 22nd. So we did ABC and furrow, and then I was wanting to see what the differences are. So the ITR is in the row, BTR is in between the row. So right in the row with the in the row of the corn, and then 15 inches away. You know, so lower, I see lower organic matter, lower phosphorus, lower, most of the macros are lower other than calcium and, well, and magnesium. But where were the micros? No, that's on the soil, or that's on the tissue sample. And I'm thinking maybe we're getting more mineralization with the biology in furrow. That plant's actually uptaking it, is my thought. You know, in between the row, we got that residue breaking down and it's actually building up. So when we plant our soybeans in between the row, seeing that there's more fertility to split the row. This is on a customer's field, those two plants that we split apart. We did soil samples where we did the ABC and where we didn't do the ABC. We saw a higher N, P, K was relatively the same, magnesium's the same, higher calcium. I kind of want to, I want to focus a little more on pushing available calcium. You know, you see in our field, we got anywhere from 2,800 parts per million to 4,000 parts per million, but yet our pH is down. I'm going to stop focusing on pH with the lime and start focusing on having more available calcium, because it's not only needed to be able to bring nutrients into the plant, it's just as important for that biology. That calcium, you know, it helps with the cell structure, the cell strength. So our little microorganisms that we can't see, calcium's just as much needed. They need calcium just as much to be able to perform their function as the plant does. So see a little more calcium in there with the tissues, but I'm seeing what I'm seeing as consistency is we're seeing more micros. And then when we were doing the bricks, we didn't get a chance to put those slides in. With the bricks, it was 3% higher where we had the ABC, and they were healthy plants. And the plants that we split apart out at the field were those same plants. Not near as much disease on the plant. There was no fungicide applied to that field. Not near as much disease, and we're seeing that with the in the ears too. You know, higher yield potential. So that's where I'm getting really excited when it comes time for planting. You know, everybody's, you know, precision planting's been getting pretty big. You know, John Deere, everybody's got these fancy electric planters. And what I don't like is the whole marketing aspect where everybody says planting's the best pass, where sometimes you hear it so much where it's just a marketing thing. But seeing you know, with it being, you know, August 15th, something that the only difference was at planting, and seeing that different of the difference in the tissue samples. You know, boron's a big deal when it comes to grain fill. Zinc's a big deal when it comes to grain fill. I know, looking through some other samples that are in this account, zinc, boron, and are always elevated, and those are, and manganese sometimes. You know, so those are the three critical nutrients. You know, and what's crazy is, you know, when we think about, you know, when I said what makes up a healthy plant, 94% of it's made out of carbon, oxygen, and hydrogen. 3% of it's nitrogen. The other 3% is everything else. So that, you know, it's kind of getting the mindset of, you know, maybe these micros really aren't that important. But looking at this data again, if it's only 0.001% of boron that makes up that plant, that boron provides a critical function that if we're short on it, you know, it's proven that, you know, putting boron in with your fungicide pass always pays. Boron on beans. James has done a lot of you know, foliar feeding at the end of the year, and it's always manganese, boron, and zinc, isn't it?
Mm.
Yeah. Well, it's always through your Snapchat feeding another shot of boron.
Boron gets in there, retire.
Yeah. Plant tissues, where this guy has fungicide versus Biomentum. You know, the fungicide, there's more NPK, you know, the Biomentum, and actually looking at those plants, the fungicide plants are greener, less disease, there's a little more pressure in the Biomentum, and this is why we're all learning about it. But I'm seeing the ears are consistent, and just like how I saw on that health imagery where that Biomentum showed up bright red, seeing that on the health map and having doubts, and then going through with the combine and being pleasantly surprised, a lot more manganese and more zinc in that. So we're seeing the grain fill, even though the plant is, you know, we're seeing a lot of our deficiencies in our plant health is reflected off of the macros. Short on nitrogen, it gets it gets yellower. I'm wondering, is that plant with that fungicide, is it keeping that nitrogen in the leaves? Is the Biomentum, you know, able to work with that plant to focus those nutrients to the ear instead of to the plant health? Any thoughts on that, Verlyn? That's just an idea that I have. So the soybean field, the history on that. Everywhere where it's dark red and orange is where we've been blessed with white mold. The reason why the west side of the field looks clean in this year, 2019, these were enlist beans. We were growing seed beans. We had enlist and extend flex beans, growing them for seed beans. Did not clean the sprayer out good enough on the first load and fried the 60 acres on the east side of the field. But they were the highest yielding beans. I mean, just stunted them. But this other side, you know, the sprayer was cleaned out by the time we were able to get through on this other side. Those beans were stunted and they out yielded because the white mold took over where the sprayer was cleaned out. 2 years later, could still see where the dicamba was sprayed. White mold, you know, under 35. So then the following year, quart of Biomentum, put it on the west side of the field. This was, so then 2 years ago, where I have it circled, you know, Decty stem borer came in, so everywhere where it's red, this was all white mold. And I did hydrogen peroxide through the pivot, and I think where I cleaned up, you know, where that plot is, I was curious on when I was learning about the nitrogen carryover, maybe I'm short on nitrogen, maybe I'm short on sulfur. I'm going to throw ATS in the pivot, or thio into the pivot, pump it on at R3, and had my pump set right to run the pivot at 100%. Started the pivot, load control got released at 11 o'clock. You know, got it scrambled, everything, got it together, went home, went to bed, woke up the next morning, the pivot only made it halfway where it should have been. But that sulfur or nitrogen did something because it yielded better there, but there was still a white mold where it was yellow. Then the following years, when I put the quart of Biomentum on, pulled off that good corn, you know, where 300 or 280 bushel average, where there was 140 pounds of nitrogen. This is the Earth Optics test on that. Wherever I sprayed that Biomentum, seeing all that history of having white mold, it's not even detected in the dirt as of last fall. And where I didn't spray the Biomentum last year, it's still there. So we'll find out if the treatments in season this year are enough to not see white mold. I haven't seen it yet, but I'm expecting if we're going to see it, it's going to be where the red squares are.
Jake, the one guy that left on me and mentioned that you can't till it in like the mouse turds and can't till it in. You have to leave on top and then treat to get them.
Yeah.
Like before you till it in, at least like grab off after.
And then some example, I threw those examples in, James, if you want to talk about that.
Yeah, he decided to not go ABCs when he mates no aminos, but he mates in Biomentum, and you know, you could see more find right here is hyphen, like picture. It's kind of good backdrop. Still learning, I guess, but it did seem like there was longer roots when you could see it visually in person. And then also my Brix level was higher in those plants. And then Clint, you did fungal the first Clint. I'm over here in the corner. You did the test for me. The Fungal-to-Bacterial was higher, also, I believe, right?
Yeah, it was, but I don't remember what. Yeah, I didn't see those this year.
And then Biomentum and Bordro on the right, and no treatment on the left.
That's the same plant.
Okay. And then Biomentum Bordro on the right, no treatment on the left. When it comes to the leaves and
Yeah, and like the Biomentum hasn't always worked for me for stopping rust, to be honest, but there's this something else in there with it to help it work better, and it was so clean. I mean, there's no disease out there. It stopped the rust, and the tar spots taken over on where it wasn't treated. That was by Lindsay. I took samples last night.
so that's all I've got. Verlyn, you want to talk?
Who's you?
Okay, let's, we just got about 10, 15 minutes here. I just want to cover a few things. There's just some take-home material for you here. On the left-hand side, this sheet here is in front. This is a field over by where I have my warehouse, where white mold is setting in already. It looks pretty ugly. It's going to take over the whole field. And so there on the left-hand corner on the bottom here, this was written by this guy right here. I'm going to pick on him. He's been using the Biomentum for 3 years. 4?
No, just 4 years ago.
Okay, that was the first time. All right. And so, you want to read it?
I don't even read it. So, 15-inch row beans, heavily manured heavily manured fields. Fighting white mold the last 15, 20 years. Been contains, tried everything, Cobra, fungicides, you name it. Nothing's worked. I went to one of Verlyn's meetings and went, all right, I'll give it a try. This was 4 years ago. Really dry, no white mold, but I was seeing something going on in my soil. So I'm like, all right, I'll give it another year. Sprayed on a quart 2 years ago. We're still dry. Wasn't seeing it, but I was seeing white mold down in the belly. So I'm like, okay, all right. And then my uncle's field was just getting hammered. 30 acres. I'm like, crap. Call Verlyn up. What do we do? He goes, all right, let's put a half a gallon on, 20, 25 gallons of water. Okay. I'm like, he goes, I'm going to do 25 out of the 30. Went out, sprayed it, stopped it. That being sold me on it right there. Stopped it completely in his tracks. The other 5 acres, I'd have to look back. I think it says it did 20 bushels?
Yeah.
And the rest of the field still did 50. It would have done more, but we had the white combine come through that year too. There's probably 20 bushels on the ground. But where the white mold was, there was no other beans on the ground in those spots. So it's
Tell
Verlyn
him-
says with the white mold it works.
Tell them what you do for beans now.
Bean, my program is we don't fight SDS yet. I don't have in-furrow on the flanner, but I do a shot with my pre-herbicides, 32 ounces. I come back post with another 32. This year, there's white mold popping up all around us, except for my fields. I cannot fight.
What kind of beans do you plant?
Pioneer enlist beans. I saw it last year. I do a hybrid test plot for the Pioneer guys. They had a bean in there that was a very high white mold bean. The sprayer blade, when they got it, it probably wasn't one that blocked by 10 bushels. Well, so I'm like, this year he goes, I don't want to sell you that bean because it just got destroyed in everybody else's. So I've got a very high white mold bean. These over 2 different fields, 45 units. I'm only buying 100,000 population, and I still can't find the white mold. And if I had to make a wild guess, I might break a couple of bean yields. Field records for me.
You start out with 100,000?
Yep, 100,000 seeds. I'll have anywhere from 90, 85,000 final stand. That's the other thing with using the biomint for start to seed. My nodes are getting closer. I mean, I can walk out there, I can find anywhere from 19, 20 nodes, 200, 250 pods as a plant. 90% are 3 bean pods or more. So, I mean, like I said, I am heavily manured. I did Haney samples this year. What do I have? 13 units or 13 parts per million, 13 to 15 on nitrates in the soil. What does that come to? Like 120 units of nitrogen? So, the stuff works. And the corn side, I'm seeing the same thing with, because I have not sprayed a fungicide in 10 years. Gone no till, but I was always fighting a little bit of stalk rot and all this stuff because I am planting right into my manure strips. But I am seeing less seedling diseases and all that with my infill with the ABC filter. Where I'm actually tempted to pick the seed treatment off next year. Or seed without it.
It's hard to do.
What's that?
It's hard to do on corn.
I know, do it early. You gotta order it early.
I wish I need NPK in there.
Is ABC in Furum?
Yeah.
NPK and Z. I'm planting right into my hog manure strips. So I'm getting all my nutrients out of that. I'm breaking down that hog manure with humic and biomentum. And the amino acids is helping feed everything.
But the fulvic and the humic, he's taking the run away too.
Yep. That took a few years to figure out which rate was best with the humic. And now I'm actually playing a little bit more with putting more fulvic in furrow. That's my whole fertility program. I'm trying to cut back on as much as I can with the hog manure. My guys can only go down to 3,200 gallons. But between the pit pumps, I'm actually found a humic source I can feed to my hogs to stabilize the manure, make my manure better. My manure samples are anywhere from 65 to 75 beyond some nitrogen per 1,000. So it's hot. Guys put on 3,200. I put out about 240 units each. That's with the ammonia and the organic mixture. And nitrates are coming back anywhere from 30 to 50 parts per million this year, which I don't like, but there's not much I can do.
That's what in nitrogen.
What's that?
30 to 50.
Oh, 30 to 50, that's anywhere from 240 to 400 units of nitrogen in those strips.
The conversion with the nitrates, if you don't check nitrate levels in your soil, what's the conversion on that? 8?
Yep.
So you take that nitrate level, if you test that times 8, and that tells you what nitrogen units you're about to. So he does that all the time because he puts a lot of manure on. So.
Any problems with phosphorus buildup?
What's that?
Any problems with having too much phosphorus out there?
Yeah, I'm getting worried. I'm afraid Diener's gonna kept me off.
Yeah.
But I don't put on any NPK, zero commercial fertilizer, just a little bit of zinc to help balance that zinc phosphorus ratio. And the other thing I'm starting to do is I'm looking at spraying dry humic over top of the soil, just to start pulling off maybe even more nutrients out of the soil, make it plant available. What you see 20 to 30 organic matters, fortified.
I messed with the humic fill out a few years ago, and the higher the CECs, the less you'll see unless you use a very lot.
I'm looking more for it with the no-till, with the manure traffic, all this, helping alleviate that compaction is my biggest concern. While you start running 10, 12,000 gallon tanks across fields, it's not good, and I want to continue being 100% no-till. Well, some guys are telling me I'm strip tilling with the hog manure, but and I saw it 2 years ago driving across the fields. This is the other thing. This was before I was doing it. I don't know if the Biomentum's doing it, but we also cover everything about with cattle manured. And every 30 feet, you could see right where that tractor manure spreader ran. 2 weeks later, all those indentions were gone. The whole ground just is sprung back up.
When you get structure to your ground, that's
Yeah.
what it does. Yep.
And the drive line is the haul items. You won't have that compaction up there first for you.
Yeah, the dragline guys still wanted to go even less, and then I can't plant right into that manure strip.
Well, you could. You just make four angles.
I don't want to do that. I like my kind of half-mile long rows.
You probably don't have to with high fertility you have.
What's that?
I wouldn't have to plant right into it with that, I wouldn't think.
No, I probably wouldn't. It's just, it matches right up, so it's like, let's do it.
He likes a challenge anyway.
Yeah, but as I've seen it 3 years ago when we're dry, I was killing plants planting right into it. And that's when I went, crap, I've got to figure something out here. And then that's started talking to my, he goes, well, try this. So I played around with about 5 different rates. My dad was getting upset because I was not getting anything done. But I kind of found that sweet spot where it works the plane right into the furrow, and I see 0 root burn now on my corn. And that's huge. Yeah. And digging roots, corn plants, what? We're cracking them off 18 inches plus deep. So,
Yep. The soil's in beautiful condition.
but this whole program is not a sprint. If you're wanting instant results, instant anything, this is, I mean, I've been no-till since 2018. Been doing this now for 4 or 5 years. I mean, it takes time. You just kind of got to stay on the page and keep going, is what I found out.
Thanks, Wade.
Yep.
This next picture is just a picture of a cornfield a mile down the road from me. We had derecho winds come through our area, and it looked like a hailstorm went through there. This was taken yesterday. And on Saturday, when I went by there, I could see a hint of it on the top of the corn. And I was gone for 2 days, Sunday, Monday, Tuesday. And I came by there yesterday, and I couldn't believe it. That corn is almost dead. That's either Goss's wilt or bacterial wilt. I'm not sure which. Probably bacterial, because it'll take it faster than Goss's actually. But that's what we got going on in our area, and so that's just for your information, let you know. Okay, I want to quickly go through this stuff on the right-hand side. After the initial page there's this sheet here on compost. And the thing I want to point out to you, I've got some stuff underlined there. But where is it? Here. The important thing I want you to realize is, you know, everybody's after carbon in the soil, but what it states here is, if you go halfway down the column, microbes are the planet's largest terrestrial stock of organic carbon and nitrogen, okay? And they are the largest, if you go down to the bottom, this carbon cycle holds an enormous potential, quicker compared to grasses, trees, or other plant biomass. There's nothing builds carbon faster than microbes. Now, if you turn that over to this one here, there's corn row. Is this what your corn rows look like with residue? We were up in South Dakota for 2 days, Clint and I were, and the producers we went to, what they complain about probably the most is all the residue. They can't get rid of the residue. And you hear that from a lot of people. This is corn rows without anything done to it. And if you look at the next page, this is corn rows that had the biomentum put on it this spring, right?
Yeah.
James?
We've been, let me think here. We've been 32, well, okay, so would have had the cuts with the Hog Manure.
Yeah.
And then it would have had 32 with the herbicide posts. And then it had 12 ounces at B7. And I think at that point there would have been 16 more at the B1 stage.
Okay.
Sorry, R1 stage.
Good. That you got to spread out over time, which is important. We're just going to talk briefly about this, but that's on corn. If you go to the back of that page, you see this one. That is in soybeans. And we saw a lot of that up in South Dakota too, didn't we, Clint? And, you know, how do you plant into soybeans with that much, or soybeans into that much residue? It's a problem, isn't it? But the biggest thing is, this is, if you take the next sheet, this is in, I think, James' field again. This is where he put the biomentum on again. And same. That's the amount of residue that's left in there.
GMO corn? Sucks.
That's your steak.
Makes a difference.
I'm not sure. There's some conventional and some non. I don't think through that.
The BT called.
Oh, yeah. And we see that. We saw that in a field here recently too, where most of the fields where it was put on, corn stalks degraded pretty well, but one of the fields didn't very well. So the reason I bring this up is because what is that residue worth to you guys? There's a dollar value associated with that. And if you look at this sheet here, the next one, harvesting crop residue, what is it worth? You see on there, clear over on the right. First of all, I've got two of them in here, this one and the next one. This one says for nitrogen, for 1 ton of corn residue, there's 37 units of nitrogen, 37 pounds of nitrogen. If you look at this sheet, and I can't find anything newer on this stuff than 2016 and 2017, not even Nebraska. Nobody publishes anything on it. But on this sheet, they have corn nitrogen in a ton of corn residue at 17 pounds, okay? And it gives you, so I don't know which one is right, actually. But the thing I like about this one, and you can plug these values in if you want to do that. It gives you NP and K there, just those three, the concentration and then the pounds per ton. They're using 40 cents a unit for nitrogen. Is that pretty close?
Yeah, everybody wishes.
Yeah. 45 cents for phosphorus and 35 cents for potassium. Now the phosphorus and potassium numbers on both these sheets are fairly close, but you can plug your own numbers in there. But what they say is where is it? If you look on the second sheet here, it says on corn, 1 ton of corn residue per 40 bushel of corn. So what'd your corn produce last year? We'll just say 200 bushel, right? What is that? That's 5 ton, right? And if you take those numbers and plug them in, you know, it's anywhere from probably $80 to $130 an acre of nutrients that you're leaving on the table if you don't get that broke down, whether it's in corn or soybeans. And here's the key. The corn plant, soybean plant, any plant, the first thing that it wants to take up is any residue breakdown, nutrients from any residue breakdown from another plant. It'll always go after that first. And if that's available, that's what it'll use. It'll use whatever you put out there next, whether it's manure or commercial fertilizer, that it'll take up next. But that's how important your plant residue is. And I know some of you guys are dealing with 2-3 years of it. And it gets to be a problem. So I just wanted to bring that up because it's important to your bottom line. Okay, we'll keep moving here. This sheet is simply, yeah.
Timing on best timing for trying to decompose the crop residue. Is it better right after harvest or let it overwinter and then trying to hit it early in the spring?
Well, that's kind of a loaded question because it depends on the temperature and the moisture in the fall. Any biological takes moisture and temperature in order for it to grow and reproduce and break stuff down, which is what the biology in the soil is meant to do. So if you get enough moisture and temperature and enough time, it will really go to work on it in the fall. But the nice part about this stuff is, like was mentioned out in the field, this stuff doesn't die. It freezes over winter, and right away in the spring it starts right up again. So you got 2 choices. You can put it on in the fall, or you can put it on in the spring. And if you're going to put it on in the spring, put it on as early as you can with your pre. Then the other nice thing about this stuff, you can put it with your herbicides. It doesn't matter, so you don't have to make another pass. So I didn't answer your question.
I guess is there problems by you said it'll freeze, so actually if a person could put it on post-harvest in an Indian summer in January, it'd actually start working then maybe freeze again.
It does. Oh yeah. Yep.
My plant is spread out right behind the combine. Sure. As soon as I get a field harvest done, but it will spray. So I can get as much activity now when it gets to cold winter, because of dormant. That if it's a wet spray, you can't get out there. Or say you get like February this year, we had some really warm days. You know, but no snow. They got to work those few days to come back and start going again until it got cold again. So the sooner the better, in my opinion. Right? That's my dream.
So that's the program we're going to go all ready with it. Just buy them in.
So what? So okay, when you put a biological down like that in the fall, there's all kinds of carbon out there. That's what it's got to break down, right? There's not enough nitrogen, and some of you guys have been to the meetings we have, but it takes carbon and nitrogen to break this stuff down. What we suggest, and what a lot of companies suggest, is you put a nitrogen source out there for one thing. So what we basically do is we use a product, basically we use the ABC, okay? Because it's got a ready source of carbon to help it grow, and the amino acids is a ready source of nitrogen, so that it's got something to get started with. Because if all it's got is the carbon out there of the corn stalks, it takes longer for it to get going. So you always want to put a carbon, and it can be sugar, it can be 32% or 28%. It doesn't matter. It doesn't have to be the ABC if you're just going to use the biology, but put a carbon and put a nitrogen source with it, and you'll be much happier. Thank you, Russ, for bringing that up. Wade?
One thing I've also been seeing, I've been doing it in the fall, so I put the ABC and then I throw in 2 pounds of AMS. But the other thing I'm also seeing is my snow melt is so much faster than all my neighbors. Because all that biology, there is certain biology that will work over winter in frozen soils. So I'll lose all my snow about a week and a half before all my neighbors. And nothing washes away, then it all goes to the ground, where towards early spring when you get those really warm days and all of a sudden all that snow melts and it's all washing away, where mine is all gone right back into the soil.
See, in that biology, it's reproducing. And I don't want to make Brittany blush, but you get warm when you have sex, right? And that's what's happening in the soil. You get all that activity going and that reproduction, and that's what happens. So, but I just had, go ahead.
Oh, say I wanted to spread spring before planting. And I wanted to start breaking down more stalks. How many weeks in advance would you need to possibly have a breakdown enough to help the plantability of the soybeans?
It goes back again to temperature and moisture. So there's no set rule on that. You can't just say, okay, I'm going to put it on today because a month from now I want that stuff gone so I can plant. It's better that it happens over the course of the growing season, because it's going to be feeding that plant all, up to this time of year. So there's not a good answer to that. Sorry.
And ask the weatherman, we need to go.
Yeah, could do that.
Can you run that through a pivot?
Yeah, absolutely.
Fall or spring view?
Yeah, absolutely.
Least amount of water you done.
Yeah, doesn't matter. You just need, actually, water with it is going to activate it faster. So moisture is what stops biological production faster than temperature.
Too much or not enough?
Not enough.
Not enough. Because last year I had about 0 breakdown, but just as we started getting some moisture and heat, that just ramped up and those corn stalks broke down this past spring.
Yep. All of a sudden, poof.
Yeah. Because that's the other thing I fight. Growing 240, 60 bushel corn, it's just 15-inch row flanner. My dad was plumbing up all the time. It just broke. Complaint. Now we've been doing this. He loves it. Never plugs up. Everything just flows right through.
It'll slice a lot easier.
Yeah, that's the other thing. It's this BT corn that rives so stinking hard now that, and I'm planting lower populations, 28,000, and my yields are still the same or better. So,
Any more questions on that? The other thing that I forgot to mention, that I think was mentioned, but if you guys, anybody have trouble with white mold in here? Yeah. Okay. If you're going to use this for white mold, we suggest 1 quart to 2 quarts. But when you put it on, if you till, it's not going to control the white mold. It's got to stay on top on the residue. If you're going to till and then put it on. But if you till, you've just wasted your time and money. Okay?
You didn't waste backwards.
Well, you didn't waste it, but I mean, you're not going to stop any white mold.
Yeah.
Okay? Forgot to mention that. This is just, so everybody says, so what's in that stuff, So this is what's in it. If you want to know, this is what's in it. It tells you there's different microhygias in there. There's no secrets here. It breaks down, solubilizes those nutrients. And if you look at number 2, 3 different saprophytic fungi, they recycle nature, what we're just talking about, but there's stuff in there that also destroys pathogenic fungi. There's cell wall borers, destroys bionematicide fungi, infects eggs and juveniles, and that's what we were talking about with the rootworm, eggs and stuff like that too. So, and then there's a bunch of different bacterial cultures in there. So, okay. We were talking about the relationship to bacterial fungal. I like to show this little slide here, and this is for you to put up on your wall so you don't forget what this whole program is about. But Dr. Ingham did this test. It shows how much bacteria in proportion to fungal is in a forest. 500 to 1200, 500 bacteria to 1200 fungal. And if you go down here where she took and put fungal on a pasture, took a pasture, split it in half, put cattle out there, where they had 675 units of bacteria to 830 of fungal, higher fungal. Remember we were talking about that earlier. 2 pounds of average daily gain, where it was 230 bacterial to 50 fungal, 1 pound. Same way, they did the same thing on corn, 180 bushels, where it was almost 50-50 like we were talking about, and 100 bushels where the bacterial was way lower than the fungal. And there's a little blip on the other side about the relationship to what fungal does and how you can tell if there's a symptom of fungal deficiency. Poor soil structure, poor calcium retention. Jake was talking about the importance of calcium, and it shows there's a little thing in there on what Dr. Ingham did with pouring calcium through bacterial-dominated soil versus 100% fungal. Retained all of it with fungal, and the rest of it just all ran through. And then the third was inadequate phosphorus uptake. And the fourth one, increasing soil diseases. This was done years ago. This is nothing new. Okay? Then I just thought we'd throw this in there. Somebody sent, who sent this picture? One of you guys in here? No. Okay, this was from another distributor. He had a customer first time this year that put the ABC in furrow, and he'd never seen anything like this. All that cobwebbing in there, and what looked like eggs in there, which are actually spores for the fungal. Yeah, and he'd never seen that before. He said somewhere the size of an egg that he was digging up. So, anyway, egg. Yeah. Yep. So he had the right stuff going on in his soil. This was his first year, so he was flabbergasted. Okay. I told you I'd keep it short. It's just some information for you to keep and review. There's no other questions. I don't have anything else, and I appreciate your attention in coming today. You can hang around and visit with, or you're free to go. There's still some sandwiches up here, so.
Any research with corn nematodes?
Okay. Wade, you want to talk to him about nematodes and the one guy up there? What happened with the numbers? Nematodes? Yeah, after your field day, we were up there by the.
That was the David that sprayed fungicide on hot balls.
No.
It's
So
in my brain.
he didn't use the aminos.
Oh, yeah, Nathan. Okay. So we left the amino acids out in the ABC program. It's like, well, let's just leave that out and let's see what happened. Well, the biomentum overloaded and burned off all his nitrogen in soil, so he ended up having to top dress nitrogen now. The biology, this is first year using it, doing Haney samples. He thought he was on the right track doing cover crops and all this, and he thought he was getting his fungal-to-bacterial balance. And first year using biomentum, and his soils were so out of whack that they're heavily bacterial to almost sterile, that the biomentum burned up everything in the soil so fast, all the urea.
It gave it
tilled.
It had the carbon.
Was it tilled?
No, he's been no-tilling 8-10 years now too.
See, it had the carbon because bacteria is 5 carbon to 1 nitrogen. Fungal is 30 carbon to 1 nitrogen. And what we're doing is we're trying to build fungal to get this whole system working. And it used the carbon, but it didn't have the amino acid there for the nitrogen. So it used what was there in the soil, and it backfired on them. There's a reason the ABC has in it what it has in it. It's a balance. So, but he was the same one that had the nematode differences, remember? They went up to 6 or 7 on one and they not on the other.
Oh, on the PFLA sample.
Yeah.
Yeah.
That's what he's asking about.
Yeah.
Yeah.
I can't remember how that went again. I don't pull that up.
It was on that same. It was on that same without the amino acids in there.
Because you get your good nematodes and bad ones. It's almost like the biomentum's helping control the bad ones in your corn.
They do.
If that makes sense.
They do.
Because I just learned like last week that Nebraska is notorious for having an overabundance of bad corn nematodes.
Yep.
And I've really never before. And so that's why I asked the question.
We're hoping to get that answer from Earth Optics this year because that is a problem. Cyst nematodes, of course, in soybeans, right? And so we're hoping to get those answers, but where I see those issues, especially cyst nematodes, is where it's a tight soil. I've for years already, I've had working with producers. How come I have that? How come that spot's got them and that spot's got them and that spot's got them? Well, you go out there with a penetrometer, you can't hardly get it in the ground. It's tight soils. So, but we hope to get some of those answers this year, but we have not specifically done anything like that.
A lot of high megs then, or there's tight soils for you?
What's that?
With a lot of high meg on the tight soils then, or?
Yeah, a lot of times it is. The one particular guy I can think of, if it rained on his yard there and then it dried up, it was just like a rock, a lot of magnesium. Yeah.
Tillage helps with that too. You got that soil, so there's no flocculation in the soil. The pack
No structures.
No structure. Yeah.
Yeah.
It packs tighter.
Yep. It does. So,
Any other questions?
You said you're putting biomentum on in fall or spring, or where you collect nitrogen and carbon. What is it? Where are you rates and what products are you?
Very much a really nice cart.
So what we're doing, because it's just what we have and what we do, so we put a humate. So in the ABC, the A is the amino acid, okay? The B is the bacteria, and the C is the carbon. The carbon is simply a liquefied humate, is all it is. And that carbon can be sugar, like I said, it can be anything. You can use your own sources. Just please use a carbon and a nitrogen source. If you're going to use sugar, if you use a half a pound of sugar, that's plenty. If you're going to use like a 28 or something, a pint is plenty. Plenty. It doesn't even have to be that much. Okay? And the Biomentum mixes with everything. Those things will not kill it, just make it grow faster. When we put ABC in totes and we ship it out that way, you know, if it gets too warm out and it sets too long, it smells like a hog pit because that stuff starts growing. It does. Yep. So just warning you. Okay.
You know what I'm saying?
Oh yeah. Oh yeah.
My wife experienced, she puts on the foliar with the Biomentum,
Yeah.
and this summer, just the rain, we didn't have time. We just didn't work. But she left it sitting in that spray jug for a month. And she opened it up, and we had, I think, sugar with it and something else she had with it. She opened it up. She goes, "It's fine."
Really?
You made the comment one time it's not going to grow too much until it's put in the soil where it's got the carbon and everything to take off with.
Yeah.
And it was, she sprayed it up.
Okay.
Around the dairy, are there any pesticides that you can mix biomentum with or that will harm the?
Well, of course, since it's 25% fungal, fungicides are very hard on it. We haven't seen a terrible lot of data. You know, if somebody wants to mix it with insecticide, I'd like to look at it under the microscope first. We spend hours under the looking through a microscope with this stuff to test it and make sure it's good. But insecticide, maybe. Fungicides, absolutely. Yeah, Wade had one guy up there that after the field day, we went out to his field and tell him what we found.
Killed off all his fungal in his soil. It is. His soil was very spongy, looked awesome, working perfect. 4 years of fungicide and killed off all the fungal in the soil. Just made the ground harder than a rock.
He would
And
tell him why
you
he
can
was putting
even
it on.
see when we dug, you could see the cord roots or where he plants winter rye, and you could find all those roots in the soil, and it was just making a compaction layer, just pressing down.
So the rest of the story is, this guy, he has cattle. He farms 80 acres, right? He works for a livestock company, and he does custom chopping. He and another guy. So in the fall, they go around and chop, and his corn gets chopped last, right? So it always gets too dry. So the agronomist said to him, put fungicide on your corn and it'll stay green longer. And it does, right? Helps for that. So after 4 years, Wade planted his corn.
I planted his corn. Mine, I was having 0 to 100 pounds of uplift, just so I didn't have sidewall compaction. Soil moisture was anywhere from, because I've got smart firmers on it, 40 to 50%. That's the other thing I'm noticing, when soil moisture is going up. His was running 30 to 35% soil moisture with smart firmers. And then I was maxing out the Delta Force on the planter. I was running 450 pounds of down pressure. It's huge. And just 4 years ago, you could walk across that farm and it was just a sponge. I mean, you could leave half-inch indentation with your foot, come back down that same path, and I find you.
That's what fungicides do.
They did the NRCS underwear deal where you bury it out there, and then within 2 weeks it was gone. So now he's having to go and play fixing just because of using fungicide.
Those underwear are still there now.
Yeah, it would be if you would bury it.
So we were up in South Dakota, and Brittany's a distributor up there, and she had somebody plant their corn this year. What did they tell you, Brittany?
They're like, oh my gosh, you have so much moisture in here. Like, he could not get over that. I've only heard it 10 times in 2 days.
And
My dad passed away last fall, but he farmed like Verlyn's talk since '84. So if that gives you an understanding.
yeah, so what she's saying is this guy is also a customer of theirs up there, and he just started using this product here last few years, 3 years, sparingly, put it that way, and he came over there and couldn't believe how much more moisture their soil had than theirs. So, but you got to remember, the more fungal and biological activity you have, microbes are 70% moisture too. And just like Clint was talking about, that's why he had wouldn't have had to run the pivot that many times, because it just holds more moisture. And when you get that porosity in there, everything just the carbon level goes up and everything else, and you hold that moisture. It's exchangeable up and down. So
Years ago, when we done the meeting to do this stuff, it was always said, these chemical companies know exactly where they're going, exactly what they're doing. If it gets you to use one thing and you got to use another thing to fix that problem, it just ends up popping all the time.
it's a vicious circle you guys are caught in. I feel sorry for you, because that's where it's at.
It's not a circle if you get out of it. Get out of that circle. I mean,
It takes courage to do that.
Yeah, it does. Nobody listens to you, so.
So, thank you guys.
So,
Yeah.
what's your story? How did you get to the point now where you get product like that?
So, back in 1976, after I got dry behind the ears after I got out of college, I started working for a co-op as a livestock nutritionist, balancing rations. Worked with a lot of dairies, and every day what you feed that cow makes a difference how much milk's in that bulk tank, right? So when you change feed from one field to the other and you got to rebalance the ration, and you start putting rock in there in the form of calcium, phosphorus, and all that, it's really hard once that milk goes down the bulk tank to get it back up with unnatural sources, so to speak, okay? And so I started looking at what was going on out in the soil. And I didn't last much longer at the co-op because I started questioning that. So I went off on my own and started doing all this work, and I was with another company originally on agronomy and stuff like that, but that's how I got, and this company said, you put our fertilizer on, you'll get the biological activity going in your soil, and you won't have to use our fertilizer in 3-4 years. That never happened, and the ground stayed hard. So I kept looking and looking, and that's how I came across the biologicals. I didn't put this biological together. I bought it from a company. I bought the rights from a company to manufacture it. They showed me how, and we reproduce it. I went to school. This Dr. Elaine Ingham, I spent a week up at her school looking through a microscope and looking at different biology, learning how to identify what was good and what was bad and all that. But I got serious about this about 15, 20 years ago. And I've been doing a lot of research and development for the last 15 years. So
You make it in Ore City?
Yeah. we
Cool,
do.
bro. You
Yep.
done a lot of work making it too.
Yeah. It's a lot of work. And the problem is, if you get a contaminant in there, it's worse, makes it worse and not better. You can kill stuff with it. So just a word of warning. I've got one guy, he's down by Sioux City. He tried to make his own a few years ago. He got burned bad. And then he called up and we had a conversation. So, but now he uses it all the time. But there's a guy in Indiana that Clint's working with. He started trying to make it 20 years ago, Clint?
He was making his own biologicals, and then one day Billy flew it with it inside.
Yeah, that was, I don't know what the heck he was doing that he blew it up, but
Windows out. He had a dust explosion. And he tried making his own liquid stuff, compost teas, stuff like that. He's big into biology. He's actually big into green lightning, and they're using it with green lightning and having excellent results. But I gotta go get trial data from him after harvest, of course. And he's into radionics, which I don't know what that all is. Verlyn knows, but you told me that if you're into that, you know a lot of stuff. He told me last week or the week before it's the best biological he's ever put his hand on. And Verlyn said that if he knows how to look at it, he'll see that. So he's a very good dude, and he's got some dang good results out there. This green lightning thing, there's a lot of negativity towards it, but you gotta do it right, and it takes special, like, good clean water to do it.
Very expensive to get into, though. Yes,
I didn't price one yet, but
$40,000 for a small unit.
you could buy used units, I guess.
Oh, you can.
Yeah, supposedly, but
But we're only making roughly 100 gallons a day. These small units. If you get into the big one, it can make up to 500 gallons a day. But that's like the unit you're probably talking about is like a 4 or 6 head, and they make a 24 head.
Oh, really?
So,
They had to deal out good sewage here.
yep. And I was just gonna ask Verlyn on this: does the pH matter on spraying like the Biomentum?
the Biomentum, if you check it with a pH meter, it's around 3.5.
No. Put it in the water, pH of the water you're talking, right?
Well, the
That's what
reason
I'm
I
getting at.
asked the reason I asked that is because when they were talking, bringing up the green lightning, there was a guy that I one of my potential customers. He did an ROI system or RO system for his water, and he tested it, and it was right at a 6 to 7. And then he puts that right into the green lightning where it's mixing it, and it changes his pH all the way down to 2.5. And he has seen the best results ever on kills on weeds, because you're when you start adding those herbicides and stuff in there, you got your not breaking them down. Yeah, it's breaking them down. It's making that plant take it up really fast, and your balance for that pH is at a 5 to 6 versus, when you put your salts in there, or your, what do you want to call it, AMS,
Yep.
it is more to like a 7 or 8.
Yep.
AMS does not lower pH.
0.2.
0.2.
It would.
4 pounds per acre.
It will. 0.2. Huh? So from
was
7 down to 6.8.
Yep. And water, I was supposed to lower it for me from FTs and lower, but 0.01.
5.5.
Tested tree water.
5.5 is where you want
your pH to get maximum uptake by any herbicide.
But we have a lot of issues with our water. So
Yeah, everybody
there's
does.
a lot.
Nobody
will
checks
show
it,
us.
though.
So do you have a product that know that?
What is the effect, if any, on hard water? Like 30 plus drain.
It doesn't bother the biology one bit. And again, it can be in a pH range from 2 all the way up to 9, 10. It doesn't matter. And the hardness of the water doesn't make any difference. It's just a carrier for the biology to get into the plant, because like what was mentioned earlier, The stuff is systemic and it goes all the way through the plant. If you spray it on, I don't care how hard your water is, it takes about 2 weeks and then you start to see things happen. If you do a tissue test or whatever, I got a producer up in North Dakota. He farms 50,000 acres up there. And I send totes of that stuff up there to him. And he said, you know, that stuff is crazy. He said, you can just about count it to the day. 2 weeks after you put it on, he said, things just take off. So, but it takes that long for that biology after it gets in the root zone and gets the exudates from the plant to start multiplying and bringing nutrients back up to the plant, and then away you go. So it's a multiplication process, because we're talking not that many ounces per acre. So, but water doesn't affect it. Chlorine will affect it somewhat, yes. It will kill some of it off, not all of it. We've used it in saltwater fish tanks to clean those up, so salt doesn't affect it either. So.
We will treat the water. You got some free water.
Yeah,
we won't go to that. Okay.
Plant helps tomatoes grow, right? What everyone says. The environmental, yeah. And the water units. My wife's got been picking tomatoes for over a month and a half. Everybody else is struggling to get tomatoes this year. Something weird's going on. Couple things I wanted to add quick for one. So if you guys are using other biologicals, one is don't ever get the mindset that more biologicals is best. There is a product that you are using that you do like and you want to try the biomentum with it, get us a sample or let us know what it is and we want to look at it. I've done it with Verlyn a couple times on some other products, just to make sure they play nice together because you don't want to antagonize each other. And then the other thing I wanted, what's that? We went through the, yeah, you can get two biologicals that will just attack each other. You do it yourself worse and get. We will do that for you if you guys have products. Second thing that nobody mentioned, but if you got any hay guys in here, we do use it as a preservative for baling. You use 1 to 2 ounces per ton of hay, and we've had guys baling 26% moisture with no problems. So if you guys are doing hay and looking for something to try, that's something to try. Brittany could tell you she used it this year too.
Yeah, Brittany, tell us about your hay experience.
Well, this year we've had a lot of our customers and neighbors helping us because it's just me and my mom now. And so there's a 4-day gap, and I'm like, okay, this is my chance. So we put all the hay down, 80 acres of hay, that's all we have. And I'm like, okay, we got till about Wednesday, and you get this up. And so the day after they cut it, I actually went with the 4-wheeler up and down every row and put biology on it. So I'm like, I'm going to do my part if you're going to get this hay up dry. And that Wednesday morning, I had 3 of the neighbors there to bale. And I'm like, if it's under 26, just keep baling, it'll be okay. And they all just like stood there. I swear it took me 45 minutes to convince them to get in the baler. I'm like, it would be okay. And then the one guy hauled it all off, and he was really unsure. We even had to call Verlyn. I'm like, yeah. They would not believe me that it was going to be fine. I'm like, I promise you. Like, let's talk to Verlyn about it. And so I finally got them convinced, and they bailed it and it started raining just as it got done. And a couple weeks later, the guy that bought it from us called me, and he's like, yeah, it's keeping really good. I'm like, good. So we've done that twice this summer. So, and then actually, I was just telling Verlyn at lunch that the same guy cut it the 3rd time now. And I had told him I was going to put it on, but I never got to it because I went on the road and stuff. And he called me Saturday, I think it was, to bail it. And I'm like, yeah, I didn't get it on. I didn't get a chance to. And he's like, oh, really? Like, all bummed about it. And I'm like, okay, good. We got it. So it was really funny to watch him question me.
I got something that's working, but it costs tons more than what you're talking for costs.
Oh, really?
I mean, $6 a ton. For magnesium.
Anchor.
Oh, I know what it is.
2 ounces. I'm on the 34% moisture and keeping it, but we got higher egg for that. So say 8 to 3/4.
What is it, Clint?
What's that?
What's 2 ounces?
2 ounces is 36 cents.
Okay.
Usually when I got weather, I helped because it didn't cost much.
Yeah.
Right. Gas people or not.
Well, what if you just spray 32 ounces? The 0 ounce of treating the soil areas? It's not
That
enough.
doesn't mean you're not going to use. No, but it moves that.
Well, no, y'all.
So I actually put it on my alfalfa. Like I have 78% alfalfa. Jake kind of talked to me and he said, well, try it. I think it'll work. So I put 16 ounces over the top. And my field, when I first started farming, I had a 7-year-old gin that he fertilized and he sprayed every year. Never had anything with fertility on it. First cutting I cut, it was on those 70 acres. I think I got 119 bales, and I went and sprayed this over the top. And after I cut that cutting, and that was after first cutting is when I sprayed it on the top. I put that on the Biomentum on 16 ounces an acre, and I had, when I cut that second cutting, I had mushrooms everywhere, and I ended up getting 170 bales versus the 190. But the only thing I couldn't do is in our area it rained every 4 days, so it just started staying wet. And after I had that cutting bale, and Verlyn's like, "Well, yeah, you could have put the Biomentum on." I was like, "Now you tell me." But no, it was one of those where, even over the top, everything stayed nice and green. My windrowers were really green all the way through. It stayed wetter that way, but is also probably trying to preserve it even when I sprayed it over the top. And right now I got my third cutting, and my third cutting was 166 bales. So it went down a little bit, and now I'm going to be getting the fourth cutting here.
How many acres?
70. Right now on those 3 cuttings, I'm averaging right at probably 4.5 to 5 tons an acre, and I got another cutting year.
Nice.
So it's you can tell, and I don't sell my bales right now because everybody knows the bale market or alfalfa market sucks. But once I do sell it, I'll know tonnage, better off. But yeah, right now I'm averaging, I'm guessing 4 to 5 tons. So, well,
I just had a couple things. Finally got my probe to connect to the network. So where we were standing, in the bare spot, pH 6.59, organic matter 4.27, 2.13% total organic carbon, 3,500 parts per million of calcium, 237 parts per million of potassium, 0.3 parts per million of nitrate, grade P1 39.77, P2 60.46, and 31 parts per million of sulfur and 6 parts per million of zinc. In the rowaways, soil pH is 6.3, organic matter 3.8, 1.89% total organic carbon, 2,900 parts per million of calcium, 230 parts per million of potassium, 14 parts per million of nitrate, 37 parts per million of P1, 56.82 parts per million of P2. So
Get the fertilizer card out there.
no, it's kind of lining up with that chart that I saw last year.
Yep.
Where we had fallow, where we kind of left it fallow, and still all good.
Yep.
So besides that phenotrometer, I want to bring up this book, the When Weeds Talk, because it really kind of opens your mind. When we see weeds in the field, it's easy to determine in hard yellow corn, nitrogen deficiency, or some sort of nutrient deficiency. When we see weeds out in the field, it's the first thing we think of is herbicide deficiency. For deficient in Roundup or Liberty.
when Verlyn pointed me towards this book, it's got a lot of interesting, lot of interesting points, and I think a lot of this book was written in the 1950s and then revised in 2013. So, like those of us that are dealing, with kochia, it loves soil deficient in calcium, very low available phosphorus, and very high potassium. It loves dry ground and low humus. But there's a benefit to kochia. I'm not saying we have to leave the weeds out there. I think it's out there for a purpose. If the conditions are right, kochia roots can go 16 feet deep in the soil. A doctor from Texas A&M animal scientist says yearling steers can gain up to 4 pounds per day in the fall by grazing it. The seeds contain 24% protein. In some instances, it may be wise to limit the amount of time animals graze it because it may contain some poisons. It is salt tolerant and attacks grasshoppers. If cut and dried before it's 20 inches tall, it can milk as little as alfalfa. So we look at that as a weed, and what we're seeing, or what I'm seeing, is when, like milkweed, for some reason, make them come back, and I look through that. It loves deficient, very low available calcium, very low available phosphorus, high potassium, very high magnesium. So like we got some fields where our magnesium calcium ratios are way off, and a couple of years ago didn't see any milkweed, and this year milkweed's booming. But milkweed, a lot of that white milk is pure calcium. That taproot goes so deep. That plant's purpose is to mine calcium that's leached out of the root zone from our crops. And when that plant finishes its life cycle, there's our calcium back on top.
That's another butterfly.
Yeah, and that's another problem.
He said he had almost 3,000 parts per million of calcium in furrow. How many pounds per acre is that?
That's 6,000. 6,000 pounds per acre. It is pH is 6.3. Now, an agronomist is going to probably tell you, let's say your pH is getting too low. He's got plenty of calcium. Just got to activate it.
So what does the 9 do or 11 do?
Oh, it's quick. It's a Band-Aid.
Okay.
Jake, pretty much most of the weeds like low calcium and low humus, right?
Mm-hmm.
High potassium.
Yeah.
Yep. High potassium. It's pretty crazy looking through. If you got some low spots where it's really wet, certain weeds, lampcore love swampy ground. You got a little bit of a swamp, lens quarter popping up. And then we'll have another year. We see those weeds. Seed alp. Palmer amaranth is one that loves low calcium. This is very low calcium, very low phosphorus, and this is available phosphorus. Very high potassium, high magnesium, high boron, low humus, low porosity, poorly degraded residue, low sticky soils, and high alluvium. And we got a dryland spot in the field where I think in was it last year, 902, the dryland? 2 years ago, weeds were just nasty. We had wide rain in the spring, and that popped up, and then the next year in the corn, we had a little drier conditions. Like these weeds have to have the right conditions, and environmentally, the biology in the soil is also driven off the environment, our weather and stuff too. But I find it interesting that certain years, Palmer amaranth can dump, 200,000 seeds. Why is it the next year you don't see, 10% of that germinate, and then 2 years later, there could be the right conditions for that plant, and it takes off.
Will the bioman do anything to the weed seed itself?
No, not directly. But if you change pH in the soil,
and change structure of the soil. Well,
Okay.
indirectly, yes. Directly, no.
I'm finding through all this that, we're not so much different than the plants. And, I used to get really bad sinus infections and, just for the sake of convenience, I'd go into town, get an antibiotic and take that antibiotic, I get through it. And then the next season comes around, come down with the sinus infection. And it was always just 2 visits to the doctor every single year. And then decided I don't want to waste the trip, I'll suffer through it. So I started suffering through it. My immune system gets built up. I haven't been to the doctor in 5-6 years, for sinus infections. I very rarely get some, and instead of being down for a month, I'm suffering for a week. So I'm kind of wondering, are we so different than, than a corn plant? If, we're dinging it with that, for hitting it with that fungicide, it's that it instantly gets better, but in the long term, is that probably more susceptible?
Good point.
Same thing with the soil. If we're dinging that soil back, then that biology in the soil can't, get the nutrients it needs to, be able to fend for itself.
Same proliferations on both. A year.
Message brought.
Thanks, guys.